Yes, hi everyone. So now I'll be discussing few important things related to the structure of protein in detail. Okay, so if you go into the basic structure of protein, okay, they can ask you some diagram based questions. It's very simple. Primary, which is something like a just like a linear structure. This is something like a linear structure. A secondary, you'll have more complicated one compared to the primary. Tertiary, okay, so you'll have more complicated one compared to the secondary, and the quaternary is the most complicated, all subunits overlapping one on another. Okay, we'll learn everything in detail in examination point of view. Okay, so these are the areas. I'll be focusing on the areas where this NEET, NBE, or all these exams and MCQs will be focusing on related to the structure of proteins. So have a look out to this diagram, because they can ask you the diagram based question like marking A, B, C. A means what type of structure, diagrammatical structure of protein is, what is A, what is B. They can ask you some diagram based questions, or they can ask you a matching like thing, giving all these structures on one side and asking you to identify what is primary, secondary and tertiary in a simple way. So coming to the primary structure, you need to focus mainly on these 2 points. That is, the linear sequence of these amino acids is the main thing that you have to focus on the primary structure. And the primary structure is mainly responsible for its function. The function of the enzyme, the biological function of the enzyme is mainly concerned with the primary structure. The secondary is very simple. The secondary, you can remember it as SS, that is the spatial orientation or the arrangement of the proteins by twistings of this polypeptide chain is called as a secondary. The twistings and all these foldings will come under the secondary structure. And secondary structure is categorized into 2 types, that is the alpha helix and the beta sheets. Okay, it is of 2 types, mainly by alpha helix compared to the beta sheets. So coming to the tertiary, you can remember with ST and T, that is 3-dimensional structure, the compact structure with the hydrophobic side chains which are present inside and hydrophilic which are present on the surface. So if you say take a tertiary structure of a protein, you will have hydrophobic inside and you will have hydrophilic on the surface. And the main function of this tertiary structure of a protein is to increase the stability of the protein. So I mean, like, before going to the quaternary, okay, there will be some bonds which are going to play a vital role to maintain the stability of this tertiary structure of the protein. And these are the bonds which the paper setters are mainly concerned. Okay, there is slight difference between the type of bonds that are present in the tertiary and the quaternary. Okay, the type of bonds that are present in the tertiary are the hydrogen bonds, one, the disulfide bonds. Next is ionic interactions, that can be electrostatic bonds, and they can be hydrophobic interactions, and a few van der Waals forces of attraction. So these are the list of the bonds which are mainly concerned with the tertiary structure of the protein. So next we'll go with the quaternary, that is the most complicated structure of the protein. So before going in detail to this quaternary structure, the main important thing that you have to focus is majority of the proteins, they may lie as in a single polypeptide, but few cases you will have multiple polypeptides, that is nothing but called as oligomeric polypeptide chain. So when you have this complicated polymers in bulk in a particular protein, okay, then you have to go for this quaternary structure. This structure of subunits, okay, is this complicated structure is maintained by the quaternary structure. And some of the proteins, they're composed of 2 or more polypeptide chain, they're referred as subunits, and the spatial orientation or the arrangement of these subunits, means multiple subunits, is called as a quaternary structure. And the bonds that are present in this quaternary structure mainly are non-covalent bonds. They are hydrogen bonds, hydrophobic interactions, and ionic bonds. So you need to know the difference between the types of the bonds that are present in the tertiary structure and the types of the bonds that are necessary to maintain the quaternary structure. And the main difference that you have to focus is quaternary structure is mainly associated with the spatial arrangement of the subunits, whereas your secondary structure is mainly associated with the spatial arrangement of the proteins. Okay, here it is subunits. Okay, in the quaternary it is subunits, whereas in the case of secondary it is the polypeptide chain or the proteins that are mainly responsible for the secondary structure. So the next important focus of the paper setters is all about the denaturation of the proteins, how the proteins are denatured. So there are a list of characteristic features that you have to make a note, you have to learn, because there are many questions related to this denaturation of proteins. So going in detail, denaturation is nothing but protein is organized because it has primary, it has secondary, it has tertiary, it has quaternary structure. So the organized protein has to be deorganized. So disorganization or deorganization of a native protein is called as a denaturation. So there will be few agents that are added to make it to destroy the protein. So these can be categorized into physical agents and the chemical agents. The physical agents can be heat, like violent shaking, X-rays, UV rays. The chemical agents can be acids, alkalines, organic solvents, salts of heavy metals, urea, salsates, silicates, detergents, and XYZ. And coming to the focus of paper setter, this is an area, this is a slide where they are mainly concerned about. So if you take the denaturation of proteins, the characteristic features, so the important aspects are clearly underlined in the red color, so you can focus on them. The native helical structure of the protein is lost, the primary structure is not lost, whereas the secondary, tertiary, and quaternary structures are lost when you go with the denaturation. The protein loses its biological activity, its ability, the biological ability of the protein is lost. Denaturation proteins become insoluble in the solvents. Okay, the viscosity increases and the surface tension decreases. There will be increased ionizability and there will be increasing in the sulfohydryl group of bonds. Denaturation proteins are easily digested when compared to the raw proteins. You can see the vegetables and everything which are like cooked means it's denatured, so they are easily digested when compared with the non-cooked products. And denaturation in general is usually irreversible process. Okay, it cannot be reversed back, but if you have a careful denaturation, sometimes the careful denaturation can be reversible, that is called as renaturalization. So the example for renaturalization is that is something called as a hemoglobin undergoes denaturation in the presence of salsates, but by removal of these, the hemoglobin by itself can renatured. Okay, so this is an exception case, so you have to remember this example. And denatured proteins cannot be crystallized. So these are the important areas where the paper setters are focusing when they ask questions related to the structures, that is primary, secondary, tertiary, quaternary. You learn the basic difference with the diagram, and followed by the characteristic features of denaturation are the most important areas where I have seen multiple questions. So please learn about these aspects and prepare your own notes. All the best. Take care. Signing off, Dr. Srikanth from Team MDS Conquer.